Welcome back. So we are now on the chapter 24, nutrition, metabolism, and energy balance. And we really started on this already in one of our lab when we discussed cellular respiration. And that will be part of this particular discussion, but it's not going to be in the same manner that we discussed it. But what's going to be in your exam is the way we discuss it. I'm just going to go through it just to remind you a few things about what we discussed. So, pretty much, guys, let's now go get the ball rolling. So, first part, we discussed what some things about nutrients, which is the substance that we have in our food that's needed for our growth, for our maintenance, for our repair, and for pretty much our overall sense of well being. Macronutrients, there are many different types of nutrients. Macronutrients refer to the three major nutrients that make up a bulk of the food that we eat, which will be your carbohydrates, your lipids, and proteins. And there are also micronutrients, which come in not so big amount, in small amounts. These are your vitamins and minerals. And water is very much required, so technically it is also a nutrient. And we have what you call essential nutrients. These are the food that must be eaten because body cannot synthesize this from any other nutrient. And about 45 to 50 nutrients are considered to be essential. The non-essential ones are also vital to life, but because our body is able to uh, form it, okay, and usually it can be converted in the liver from something else into the nutrient, okay, into the one that is needed, then it's not really essential. And the energy value is measured in kilocalories, okay, and a calorie is amount of heat energy needed to raise the temperature of about 1 kilogram water by 1 degree Celsius. And one dietary calorie, which we call them, is equivalent to 1 kilocalorie. So here's actually the new plate, that is the guideline that's represented as portions of a dinner plate by the USDA. We do not anymore follow the food pyramid. Now, the food groups here are represented by your fruits, your vegetables, your grains, your protein, and your dairy, which is actually pretty much right just around there. So in the basic dietary principle, you eat only what you need. You eat less overall. Eat plenty of fruits, vegetables, and whole grain, and avoid junk food. Pretty much that's the thing here. So let's take a look at the role of our macronutrients, the carbohydrates, the lipids, and the proteins. You can see the sources of the carbohydrates right here. They are from plants, such as starchy food, complex carbohydrates, okay, are found in grains and vegetables. And then you have the sugars, the mono and the disaccharides in fruit, sugar cane, beets, honey, and milk. The insoluble fiber, okay, cellulose in vegetable, they provide the very important roughage. And soluble fiber, pectin in apples, the citrus fruits, they reduce blood cholesterol level. And you have also like a small amount of carbohydrate, the sugar in milk, lactose, and the glycogen in meats. What are the uses of carbohydrates in the body? Well, it's a fuel. It is actually used by the cells to make ATP. And we have discussed this already in our cellular metabolism a couple of weeks ago. And some cells use fat for energy. Okay, and neurons and red blood cells entirely rely on glucose. Neurons die quickly without your glucose. And excess glucose, if it is too much in the body, is not really good. It's converted into fat Well, it's also converted into glycogen and stored. Okay, that's going to be our stored like animal starch. Fructose and galactose are converted to glucose by the liver before they enter circulation. So the recommended dietary intake is about 45 to 65% of the total calories. They should pretty much mostly be made up of carbohydrates, the whole grains, and the vegetables. Simple carbohydrates such as your monosaccharides and your disaccharides should be limited. And high amounts of sugar really can lead to obesity as well as nutritional deficiency. So stay away from them. The American diet, though, is 46% carbohydrates, and that include rice, pasta, or bread. And they tend to because these tend to be inexpensive. So the higher percentages are actually seen in the low income groups. So this is actually the summary of your carbohydrate, lipid, and protein nutrients and recommended daily allowance. It's a cheat sheet for you to really browse into, get into during your studies. So let's now get into the second part, which is the lipids. Where do we get them? Well, these are the neutral fats. These are the fats that we pretty much don't like. They're the most abundant form. In animals, in our food, you find them in saturated fats, in meat, the dairy food, the tropical oils, the hydrogenated oil. Find them also in unsaturated fats, in seeds, nuts, olives, okay, most especially vegetable oil. And cholesterol pretty much is found in egg yolk, meat, organ meats, shellfish, and meat products, which pretty much is something that we should avoid because we do not really need it because our liver makes about 85% of our cholesterol. The rest of them really is coming from our diet, which we do not really need because we produce it anyway. So liver converts some fatty acid into others. Okay, there are two essential fatty acids that it cannot synthesize, and that's the linoleic acid, okay, which is an omega-6 fatty acid, and is a component of lecithin. Then the linolenic acid, an omega-3 fatty acid. And both of them are found in vegetable oils. And that's why if you pretty much take one of those uh, pretty much like oil that is rich in the omega family. Usually, the omega family is that of the six and the three because these are essential and we do not produce it. So it is essential that we get it from our diet. Okay. And pretty much the omega 6 is actually a pro inflammatory product. The omega 3, which is actually the one that is the good part is actually an anti-inflammatory product. What are the uses of lipids? Well, first of all, your adipose tissue protects us. It's an insulation. It's for fuel storage. And then you have also the phospholipids. It's actually one type of lipid. It's essential in myelin sheaths and all cell membranes. We are made up of bilayer phospholipids. And that's actually the essential component, which is pretty much like probably the most abundant of all the lipid in the human body is the phospholipid because every single cell of our body is covered by bilayer phospholipids. Then you have cholesterol. It stabilizes membranes, which is a precursor of bile salts, steroid hormones. And then you have the prostaglandin, which is used for smooth muscle contraction, for control of blood pressure and inflammation. It's an inflammatory chemical. And you got a major fuel for hepatocytes too, the lipids and skeletal muscles. And of course, they help absorb fat-soluble vitamins. So your fats in our daily requirement should represent about 20 to 35% of our calorie intake. And our saturated fats, such as butter, okay, and bacon, the fat in bacon should be limited to 10% or less of total fat intake. Cholesterol, I'm telling you, and I'll repeat myself, is not required in the diet at all. Our own liver produces cholesterol. Intake, therefore, should be kept as low as possible, especially for those with high total blood cholesterol levels, as this pretty much has been associated already with cardiovascular diseases. So where do you get them? Well, you get them in animal products such as your eggs, milk, fish, most of the meat, and vegetable also like soybeans. And these are considered to be complete proteins, and all of them will carry the needed essential amino acids. Now, legumes, nuts, and cereals would contain incomplete proteins. They lack some essential amino acid. Legumes and cereal grains together contain all the essential amino acids. So, vegetarians or vegans are very careful in choosing the vegetables that they are eating. They want to make sure that they are getting all 20 amino acid that is required for our protein synthesis. And here's an example of our total protein needs. These are the essential amino acids from tryptophan, methionine, valine, threonine, phenylalanine, etc., all the way down to histidine and arginine for infants. And unfortunately, okay, these are the only things also that are available in corn and in beans, the one in blue. Okay, so pretty much if you put them together, you will already have, if you're an adult and if you're not a baby, then you know that between these two, you already have the essential amino acids. Now, there are 20 amino acids, and there's only like a few here that's actually essential, which means our body cannot produce it, and we essentially must have it in our diet. The rest of the amino acids are non-essentials, and they are pretty much like not essential because our body can produce it, our liver can produce it. So pretty much proteins. Next one is proteins. Well, we're still in the amino acids. Okay, so we continue it. Uh, uh, the structural materials pretty much are proteins, part of the structure of the cell. And you find it in keratin, collagen, elastin, muscles, etc. And they are also functional molecules. The globular proteins, such as your antibodies, your enzymes, your hormones. Okay. And pretty much there are three factors that help determine whether amino acids are used to synthesize proteins or burned as fuel. Just think of the all or none rule. If all the amino acids that are needed must be is actually present, then proteins will be synthesized. However, if not all of them are present, then amino acids that are not going to be synthesized, and there not going to be any protein that's going to be synthesized, but the amino acids that are there are used for energy. So, protein is used as a fuel if there's insufficient carbohydrate or fat. Okay, and pretty much protein gets to be our next in line fuel. So, uh, for hormonal controls, anabolic hormones such as your growth hormones, your sex hormones, they do accelerate protein synthesis. These are exactly how some proteins are produced. The adrenal glucocorticoids are released during stress, and they promote protein breakdown and conversion of amino acid into glucose. So nitrogen balance, it's a homeostatic state where the rate of protein synthesis equals the rate of protein breakdown or loss. So you are in balance if the protein that you're taking in is actually equivalent to the protein that you're breaking down. Okay, so you are just in balance. However, you have a nitrogen balance whenever the synthesis would exceed breakdown. And usually these are normal in children, in pregnant women, and in pretty much like instances where there's tissue repair. Negative nitrogen balance is when breakdown exceeds synthesis. And this is example whenever you are in a stress situation, okay, burns, infection, injury, or you have poor dietary proteins and starvation. When your nitrogen is low and you have low protein, you have negative nitrogen balance. Positive nitrogen balance is when you are synthesizing a lot of them more than you're breaking it down. And children usually have this positive nitrogen balance, pregnant women, and tissue repair. So, the dietary requirement would reflect the age, the size of the person, the metabolic rate, and nitrogen balance. And there's always a rule of thumb: the daily intake of about 0.8 gram per kilogram of body weight is actually what you should consider. So you don't go beyond that, okay? If you're going to be like always every day eating like a whole chunk of steak, that's gonna make you gain weight. An American diet provides pretty much more than what is really needed, and that's why there's a lot of obesity in among the Americans. And here's a summary of carbohydrates, lipids, and protein nutrients. Just make sure that you know that it is available to you in this particular video, and you can take a look at it as part of your review for your next exam. So the role of vitamins and minerals. These are organic compounds that are crucial in helping the body use nutrients. And most of these are functioning as coenzymes. And most must be ingested, except for vitamin D, which is actually there's a precursor in the skin that gets converted with the help of the sun, okay, into active vitamin D. Okay, some B and K vitamins are synthesized by the intestinal bacteria, and then the beta carotene from carrots is converted in the body to vitamin A. Okay, there's no one food group, pretty much. Contains all vitamins, so they're all scattered in different food groups. And there are two types of vitamins based on their solubility in water. The water-soluble vitamins will be your B complex and your C, and your B12. Pretty much the absorption is where it requires intrinsic factor, and they are not stored in the body. Okay, and pretty much anything that is not used is excreted within 1 hour. And then the fat-soluble vitamins, this will be your ADEK, ADEK. They are absorbed with lipid digestion products. They are stored in the body except for vitamin K. And pretty much excessive consumption is actually going to cause a lot of health problems. So, dangerous free radicals are generated during normal metabolism. We do that all the time. And so we have antioxidants. These would prevent the generation of dangerous free radicals. So your vitamin C or E and A and the mineral selenium are the antioxidants, and they neutralize these free rads. Broccoli, cauliflower, Brussels sprouts are good sources of vitamin A and C. There's a note here that says, megadoses of vitamins are useless and they actually cause serious health problems depending on the vitamin involved. You do not need megadoses of vitamins. All right, so here is again a cheat sheet, a table of a list of vitamins, the major dietary sources, the major functions in the body, and pretty much the symptoms of when you have extreme excess or extreme deficiency. Take note, make sure that you study this table. Minerals. There are 7 minerals required in moderate amounts: calcium, phosphorus, potassium, sulfur, chlorine, sodium, and magnesium. And all other minerals are only required in trace amounts. Now, they work with nutrients to ensure proper body functioning, and the uptake and excretion are balanced in order to prevent toxic overload. So, For instance, how does our body use them? Well, calcium, phosphorus, and magnesium salts are the ones that keeps our bone hard. Iron is essential for oxygen binding to hemoglobin. Iodine is necessary for thyroid hormone synthesis. Sodium and chloride are essential electrolytes in the blood. And you find them in green leafy vegetables, in legumes, in milk, and some metals. And again, you got the list of minerals in the body, which I think will be helpful in your study. So I'm including them for you to browse on. Now let's get into part two, which is on metabolism. Metabolism is the sum, it's the overall, it's a sum of all of the biochemical reactions inside the cell involving the food that we are eating, the nutrients. And there are two types of, metabolism. That's catabolism and anabolism. Anabolism is the synthesis of large molecules from smaller ones. For instance, the synthesis of protein from amino acids, the synthesis of carbohydrates, complex carbohydrates from monosaccharides. Okay, these are what you call anabolism. Catabolism will be the hydrolysis, the reverse of the dehydration synthesis of these units, structural units. So when you have a huge macromolecule and you hydrolyze them into simpler ones, like you break down your proteins into amino acid, you break down your starch or glycogen into their simpler form, their monosaccharides and disaccharides. That's actually what you call catabolism. So cellular respiration, we've done this a few days ago, is a catabolic breakdown of food fuels whereby energy from food is captured, okay, and pretty much they are the ones that are involved in the production of ATP. The goal of cellular respiration is really to trap chemical energy in ATP. Energy can also be stored in glycogen and in fats, which can be broken down later. Now, the word phosphorylation pretty much is when enzymes shift high energy phosphate groups of ATP to other molecules. Phosphorylated molecules become activated to perform cellular products. And like for instance, with your ATP, okay, the low version, the less energy ADP or adenosine diphosphate will only have two phosphate groups. You add one more ATP by phosphorylation and you are transforming it into ATP. And then that phosphate, that addition of phosphate group, that addition of one high energy bond to bring in the third phosphate group pretty much will energize ATP and it will now have more energy than just ADP. So there are three stages, okay, in processing nutrients. First one, stage one, you have digestion, absorption, and transport to tissues. Stage two, you have the cellular processing in the cells in the cytoplasm. It involves synthesis of lipids, proteins, and glycogen, or catabolism, such as breakdown of glucose, which is glycolysis, into pyruvic acid, an essential acetyl coenzyme. Stage three is oxidative breakdown of intermediates into carbon dioxide, water, ATP, and they all occur in the mitochondria. This is actually the three stages that I have already discussed with you, and I'm just giving you a little bit of a synopsis of our previous discussion of what happened in cellular respiration. So, cellular respiration consists of glycolysis of stage 2 and of stage 3. So, here are the three stage nutrients here in this particular order here are digested into absorbable units, absorbed into the blood and transported into the tissues. And then in the tissue cells, okay, for instance, your broken down amino acids, okay, could pretty much like, there's going to be like the two It could be either be anabolically formed into proteins, okay, uh, where the nutrients that you've taken in pretty much are built into macromolecules such as your protein, your glycogen, and your triglyceride. And in catabolism, pretty much, let me just give myself something to work on here. And in catabolism, the nutrients that are actually like partially broken down, okay? And then glycolysis is the major catabolic pathway, and that ends in pyruvic acid. So right here in your carbohydrates, you're showing here already that there is glycolysis happening here, which is a breakdown of glucose, and 2-pyruvic acid has been formed. And part of it, you are actually, because when you are grooming your pyruvic acid to get into the Krebs cycle, um, remember that acetyl is the only one that is actually allowed to get into the citric acid, and the pyruvic acid has one extra carbon in it. So it will be groomed by the coenzyme A, with the help of the coenzyme A, and in the process, you are releasing one carbon dioxide per pyruvic acid. So the acetyls now will be released to join in with the oxaloacetate right here in the citric acid cycle. In the end, you are all again producing the rest of the carbon dioxide. Okay, remember that in your sugar, which is C6H12O6, you break down for the first two pyruvates that you are grooming two carbon dioxide here. You're going to be releasing two carbon dioxide, and the rest of the four carbon dioxides are going to be formed as what we call this, the citric acid acyl goes on two cycles and pulls both acetyl that enter the Krebs cycle, and in the process forms four more carbon dioxide, and you have six nicotinamide adenine dinucleotide reduced form, and then your FADH2 also, and then two pieces of ATP. And then all of them, okay, the high energy molecule, the ones that are produced in glycolysis, in the transition reaction, in the citric acid cycle, all of the NADH and the FADH2 are now going to be used here in the oxidative phosphorylation right through the electron transport chain, and the process will be producing enough ATP. All in all, from the beginning all the way to the end, the ATP that's going to be produced here will be 38 pieces of ATP. Okay, so let's take a look at some reactions, the redox reactions, and the role of coenzymes. Oxidation reactions involve the gain of oxygen or loss of hydrogen atoms in any reaction that's happening in the body. You call that oxidation. Now, oxidation reduction or redox reaction pretty much is a combination of both. Oxidized substances here lose electrons and energy. Reduced substances, on the other hand, would gain electrons and energy. Redox reactions are catalyzed by enzymes and usually require V vitamin coenzymes. And for instance, your dehydrogenases catalyze removal of hydrogen ions. Your oxidases would catalyze the transfer of oxygen. Okay? So there are two very important enzymes that I have mentioned in our cellular respiration. That will be the nicotinamide adenine dinucleotide and the flavin adenine dinucleotide. Okay, and these are the reduced forms that I have mentioned, not the oxidized form. Okay, so the synthesis of ATP is actually two ways. Okay, two mechanisms are involved here. One of them is what you call the substrate level phosphorylation, where high energy phosphate groups are directly transferred to the ADP. So ADP plus P is ATP. That's pretty much the simplest, okay? That's what you call substrate level phosphorylation. And they occur twice in glycolysis and once in the Krebs cycle. And that's why these two have, raw ATP being produced, not only the high energy molecule. And here it is, guys. It is going to be, um, what do you call this, helped by an enzyme, catalyzed by an enzyme. ADP plus P, okay, your substrate, um, pretty much on the substrate level, ADP plus P will form ATP. Then you have the second type, which is called oxidative phosphorylation. And I mentioned this to you in the last time that we have spoken. We did a lecture, and I showed you that in the oxidative phosphorylation, um, more complex. It's actually more. It's the use of the high energy complex molecules NADH and FADH2 to form most of the ATP from a chemoosmotic process, which is pretty much the very complex movement of substances across the membrane to chemical reaction. And the end molecule that would accept the final electron acceptor on the electron transport chain in the chemoosmosis is actually oxygen. Oxygen here plays a major role, and that's the reason why we need to breathe in oxygen. Oxygen is important in the production of ATP. It's a final electron acceptor on the electron transport chain. Okay, and here you have pretty much like only a very short, showing a very short part of the oxidative phosphorylation, and where it shows you the electron pump, the electron transport protein pump. Okay, this is actually the one that pumps the protons, creating a proton. Where pretty much your electron transport chain is the one that pumps the protons. Now, this is your ATP synthase. The purple one is the ATP synthase. It uses the energy of the proton gradient, this proton gradient, which is accumulating right there on one side of the mitochondrial membrane. Okay, it is actually using this particular gradient to bind the phosphate groups to ADP to form ATP. And this occurs only in the matrix of the mitochondria. Okay, so when glucose enters the cell, it is phosphorylated, that is actually in glycolysis, to glucose 6-phosphate. So most cells lack enzymes for reverse reaction. So glucose becomes trapped inside the cell. Now, only cells in the intestine, in the liver, in the kidney and liver can reverse reaction and release glucose back into being glucose. So it keeps the intracellular glucose concentration low, which ensures that there is going to be continued glucose entry. So glucose is oxidized, and complete glucose metabolism would require 3 pathways. And I mentioned that to you in the previous discussion we had, and this is glycolysis, the Krebs cycle, and the electron transport chain. And for the oxidation of glucose, for every 1 molecule of glucose, we are expecting 38 pieces. Well, roughly 38 pieces of ATP. But in reality, we do not really produce that much ATP because a lot of the ATP that is also being produced there is being invested as fuels in the process. Okay, and here is actually a bird's eye view, a full view of how cellular respiration is happening. And glycolysis right here, which happens in the cytoplasm, breaks down each glucose into 2 molecules of pyruvic acid. The pyruvic acid now is going to be groomed and will be groomed to enter the citric acid. Pyruvic acid has only 3 carbon, but then during the grooming process, then acetyl coenzyme breaks one of them, and so the acetyl is now going to enter the citric acid cycle. In the process, ATP is going to be produced here. And here ATP is also going to be produced. If it's going to be per glucose, you are producing 2 ATP here and another 2 ATP here. Then during the process, you are producing high energy molecules between glycolysis, the transition reaction, and your citric acid cycle. There's actually like tons of NADH and a couple of FADH2 will be formed. And then these now, through a series of chemical energy, will now be going into the electron transport chain and oxidative phosphorylation, uh, producing more ATP. Okay, so these high energy rich electrons is picked up here, okay, by coenzymes and are transferred to the electron transport chain that is actually built into the mitochondrial matrix and mitochondrial membrane. The electron transport chain carries out the oxidative phosphorylation, which ultimately will join the rest of the other 34 more ATP because you produce 2 here and then 2 here, and then 34 more is going to be produced here. Roughly, that's actually what theoretically is going to happen if you're going to be counting it. Because for every one NADH is actually helping the cell produce 3 more ATP, and FADH2 helps the cell produce 2 ATP. Okay, so this is just a more complex view of it, but it's the same banana. All right. Now, and that's actually your ATP synthase, beautiful ATP synthase. Now, let's take a look at some homeostatic imbalance. Cyanide. This is a gas that is used in gas chambers. It disrupts oxidative phosphorylation by binding to the cytochrome oxidase. It blocks the electron flow from complex 4, okay, to oxygen. There are also poisons which we call uncouplers. An example of this is DNP or dinitrophenol. They abolish a proton gradient by making inner mitochondrial membrane permeable to hydrogen ion. Although the electron transport chain continues to deliver electrons to oxygen at a very furious pace, the oxygen consumption rises and no ATP is made. So, pretty much, energy use at rest would average about 100 kilocalories per hour, which is an equivalent to about 116 watts. That's slightly more than a light bulb uses. It places a huge demand on the mitochondria for energy production. This is really very high. Now, of the 686 kilocalories in 1 mole of glucose, and it's exactly how one mole of glucose provides us 686 kilocalories, 262 kilocalories are captured in ATP, while the rest of them is lost as heat energy, which means our cellular respiration translates into 38% efficiency if you are going to view it that way. Well, that's not so bad considering that machines are only around 10 to 30% efficient. We do have a good, energy conversion in our cells. Okay, and this is again one of them. Explanation. All right. Now we get into some reactions that are happening or that are occurring in our organs, for instance, in the liver. And pretty much these reactions will pretty much give you an idea of how nutrients are processed in the body. Let's start with glycogenesis and glycogenolysis. These are processes that are contrasting. Their work is contrary to each other. Okay. Usually the cells cannot store large amounts of ATP. You cannot store large amounts of ATP, and therefore ATP, if you have a rising level of ATP, that would inhibit glucose catabolism and promote glycogen or fat formation. Okay, we cannot store large amounts of ATP. What we store is actually glycogen. So, how is glycogen formed? Well, they're formed in glycogenesis. It can be formed with excess glucose, okay, and it's catalyzed by glycogen synthase. Glucose is converted to glucose 6 phosphate, and then glucose 6 phosphate right here, okay, then converted to the isomer glucose 1 phosphate, and a terminal phosphate is removed as enzyme catalyzes the attachment of glucose to the growing chain. They mostly occur in the liver and in the skeletal muscle. This is exactly what happens in glycogenesis. Then glycogenolysis, okay, is the breakdown of glycogen to release glucose that's stimulated by low blood glucose level. Okay, here the enzyme splits and phosphorylates terminal glucose or glycogen. Okay, it forms glucose 1 phosphate, which is converted into glucose 6 phosphates, which then can enter glycolysis in that cell. Liver cells, as well as some kidney intestinal cells, also contain enzyme glucose 6 phosphate, producing free glucose. Glucose can now enter the bloodstream to be used by other cells. So, gluconeogenesis is the process of forming glucose from non-carbohydrate sources. And examples of this would be non-carbohydrate, would be protein, okay? And usually we thought that glycogen and glucose can only be coming from carbohydrates. No, you can pretty much form them from non-carbohydrate sources, okay? Glucose can be formed from glycerol and amino acid, which means fat, okay, and protein. When glucose levels drop, this protects us against damaging effect of low blood glucose, which is hypoglycemia. And glucose really is very important for the nervous system. We depend on that. Our brain depends on that. So here's a quick summary of carbohydrate reactions. First one is glycolysis, converts glucose to pyruvic acid. Then your glycogenesis polymerizes glucose to form glycogen. Glycogenolysis, this would be the hydrolysis of glycogen back to glucose monomers. Then gluconeogenesis, the formation of glucose from non-carbohydrate precursors. Let's now go to lipid metabolism. Fats, lipids, provide a greater energy yield from glucose or protein catabolism. Now, fat catabolism yields 9 kilocalories per gram versus the 4 kilocalories per gram carbohydrates. So, lipid is really more calorigenic. Now, most products of fat digestion are transported in the lymph as chylomicrons, and they are hydrolyzed by endothelial cells into fatty acids and glycerol. So, let's take a look at the oxidation of glycerol and fatty acid. Now, remember that only triglycerides, which is your animal fat, are routinely oxidized for energy. Now, two building blocks of triglycerides are oxidized separately. Your glycerol. Glycerol is broken down into glyceraldehyde 3 phosphate, and which is the same as in glycolysis, which is then entering the citric acid cycle. Okay? So your glycerol is right here, broken down by glycolysis, and it enters into the citric acid cycle. Now, ATP yield is roughly half of that of glucose because the glyceraldehyde is only half glucose. It yields about 15 ATP for every glycerol. Then the fatty acid, the other side, undergoes beta oxidation in the mitochondria. Fatty acid chains are broken into 2 carbon acidic fragments and coenzyme FAD and NAD are reduced in the process. And pretty much during the process of reduction, you are actually producing high energy NADH and FADH too. The reduced coenzymes will now be entering the electron transport chain, and this is referred to as the beta oxidation because two carbons are broken off fatty acid chain, allowing a third position carbon to be oxidized. Now, lipogenesis, the fat formation. How we actually form our fat belly. Lipogenesis. Triglyceride synthesis that occurs when cellular ATP and glucose levels are high. So, eat a lot of sugar, okay, you are increasing your chances of increasing lipogenesis. Dietary glycerol and fatty acids are not needed for energy, okay, that are not needed for energy are stored as triglycerides and they become part and parcel of your pot belly. 50% are stored in the adipose tissue and the others are deposited in other areas. Glucose is easily converted to fat because the acetyl coenzyme is an intermediate in glucose catabolism and the starting point for fatty acid synthesis. So lipolysis is a breakdown of stored fat. So this is what we are expecting to do when we want to lose weight. Okay, they are broken down into glycerol and fatty acid, which is the reverse of lipogenesis. Fatty acids are actually preferred by the liver, by the cardiac muscle, by the resting skeletal muscle for fuel. Lipolysis, though, is accelerated when carbohydrate intake is inadequate. So you are pretty much like encouraging the breakdown of fat when you are eating low carb, where you are in a low carb diet. Beta oxidation of released fatty acid results in production of large amounts of acetyl coenzyme. And acetyl coenzyme can enter the Krebs cycle only if enough intermediates, which is your oxaloacetates, are available. If the intermediates are not available, the acetyl coenzyme will actually accumulate. And your accumulated, too much of this acetyl coenzyme can be converted by ketogenesis to ketone bodies. And ketone bodies include your acetoacetic acid, your beta hydroxybutyric acid, as well as acetone. So, this is a quick summary of lipid reactions. There's beta oxidation, which is conversion of fatty acid into acetyl coenzyme. There's lipolysis, which is the breakdown of lipids to fatty acids and glycerol, and then lipogenesis, which is the formation of lipids from acetyl coenzyme and glyceraldehyde 3-phosphates. So, some homeostatic imbalances, accumulation of ketone bodies in the blood can lead to ketosis, which is very common in starvation or dieting, that is starvation dieting, or diabetes mellitus. Ketone bodies here are also excreted in the urine. Ketones are acidic, so a buildup of these molecules can lead to metabolic acidosis, which can dangerously lower the pH values of the blood. Here you pretty much like have patient's breath that can smell fruity from vaporizing acetone. Also, breathing becomes rapid as the lungs try to release carbon dioxide to raise the pH. Lipids play many roles in structural materials, and one of them is your phospholipid, pretty much is used for the cell membrane. Cholesterol is used for cell membrane as well. And in addition, the liver synthesizes and transports lipoproteins for cholesterol and fats. It synthesizes cholesterol from acetyl coenzyme A and uses cholesterol to form bile salts. Okay, so much with lipids. Let's now get into protein metabolism. We're winding down. This is the last of the macromolecules. Now, take note that proteins deteriorate So they need to be continually broken down and they need to be continually replaced. Amino acids are recycled to new proteins, okay, or different compounds. Proteins are not stored in the body. When dietary proteins are in excess, amino acids are oxidized for energy or is actually converted to fat for starch. So eating still a lot of those heavy big steak is not really going to build you muscles, it's going to build you fat. So the goal in the degradation of amino acid, the goal is to produce molecules that can be used for energy in a citric acid cycle or can be converted into glucose. So first step in amino acid breakdown is deamination. You try to remove or get rid of the amine group, okay? And then converts it into pyruvic acid, okay? Or 1 keto acid intermediate in your citric acid cycle. Now, there are three events of the amino acid degradation. One is your transamination, oxidative deamination, and your keto acid modification. Transamination happens when many amino acids transfer their amine group to the alpha ketoglutarate acid, transforming it into the glutamic acid. As a result, the original amino acid becomes a keto acid, and the glutamic acid here is an intermediate keto acid of the citric acid cycle. But this reaction here is reversible. So, oxidative deamination, still removal of the amine group. Remember that here you are trying to remove the amine group to pretty much like degrade the amino acid. In oxidative deamination, the liver, okay, in the liver, the amine group is actually removed as ammonia. Okay, here the alpha ketoglutarate acid is regenerated. Then ammonia recombines with carbon dioxide to form urea and water. And urea is released to blood and excreted in the urine. This urea cycle helps get rid of toxic ammonia. Now, there is also such thing as your keto acid modification. Keto acids formed from transamination are altered to produce metabolites that can enter the citric acid cycle. Major metabolites that are produced will be your pyruvic acid, your acetyl coenzyme, your alpha ketoglutarate acid, or your oxaloacetate. Glycolysis reactions here are reversible, so the pyruvate and the metabolites that are formed can be reconverted back to glucose, and they contribute to gluconeogenesis. And pretty much these are the three processes that occur when amino acids are utilized for energy. You have your transamination, your keto acid modification, and your oxidative deamination. This is just a summary pretty much of what happens, okay, and when we are using amino acids for energy. So a quick summary of amino acid breakdown. Transamination is the transfer of an amine group from an amino acid to an alpha ketoglutarate acid, regenerating the glutamic acid. And oxidative deamination removes an amine group as ammonia from glutamic acid and regenerates the alpha ketoglutarate acid. That's it, guys. And we're now going to get into the next PowerPoint.